This is for Biology 223 Genetics. I'm going to talk about two papers. These relate to chapters 11, 12, 13 to some extent, and also generally some biological phenomena. Here, what's interesting is that I want to talk about two papers, two reports published back to back in Nature in 2004. The two different research groups were working and came up with findings near at the same time, so the journal publishes them next to each other. These are about discovery of the enzymes that recycle vitamin K. I'm going to start, so I'm going to start with this one, even though it's the second. So this one is page 541. This one is from page 537, but let me start with it. Excuse me. So title is Identification of the Gene for Vitamin K Epoxide Reductase. So vitamin K is used in gamma carboxylation of some amino acids, specifically some of the proteins that are involved in blood coagulation. And so we need a relatively small amount in our bodies, and when we get some vitamin K, whether from the microbes in our gut or from an injection at birth, which is common in industrialized countries, it is recycled. It's used up and it needs to be reduced. It's oxidized, needs to be reduced, and the enzymes that caused it to be used or to be recycled to be productive, so it would be at this state and maybe gets reduced, whatever way it is. So the enzymes involved in recycling it so that it could be, sorry, that should be I did that, so that it can be used again and again after, and as a proper coenzyme in catalysis. They weren't known. So identification of the gene for vitamin K epoxide reductase. So that's necessary for recycling. Vitamin K epoxide reductase, VKOR, is a target of warfarin, the most widely prescribed anticoagulant for thromboembolic disorders. So there's quite a bit of a story behind this warfarin. I have the structure here. It was discovered because cattle who were eating fodder that had some, I think it was tansy or something in it, that fodder that is silage, it ferments, and there's some coumarin compound derivative that formed, and the cattle bled, and I think it was University of Wisconsin scientists recognized it, got a patent on it, earned a lot of money for the university, so it's become a very important drug. So this warfarin then works by inhibiting the reductase that recycles vitamin K that's involved in making the blood coagulation enzymes active so the cascade can occur and blood can clot. So this and then is an inhibitor of the reductase and then causes blood to be thinned, as it were. And you will talk to physicians and they'll talk about the balance of keeping somebody's blood thin so they don't have an embolism and a stroke because there's reduced blood flow, ischemia, and thrombosis, or the balance between the blood being too thin and then hemorrhage and bleeding, which can cause a different kind of stroke and different kinds of problems. So it's the most widely prescribed anticoagulant that's very many people. Although estimated to prevent 20 strokes per induced bleeding episode, that's then telling us. So the first sentence is the general phenomenon. This is the fact, and then it's telling us something else of interest, that the balance is 20 strokes are prevented for every bleeding. So the balance is pretty good, 20 to 1. Warfarin is underused because of difficulty in controlling dosage and fear of inducing bleeding. So it is not as prescribed as widely as possible. That's the more specific phenomenon. Although identified in 1974, the enzyme has yet to be purified or is gene identified. So the warfarin, the gene that we knew that vitamin K was identified before that. We knew that it must be recycled, but the enzyme hadn't been purified, and its gene wasn't identified. And its gene wasn't identified, then it limits a lot of things that can be done. A positional cloning approach, so this is back from those chapters 7, 8, 9. Positional cloning approach has become possible after mapping warfarin resistance to rat chromosome 1. Now you might wonder why rat chromosome. It's because warfarin is also a widely prescribed rat poison. It causes bleeding. So it's used as a rat poison to protect granaries, and then the rats have developed resistance to it. That resistance was relatively easily mapped. It was identified. Then rat chromosome 1 as a model organism, easy, and a vitamin K dependent protein deficiencies in this synthetic region of human chromosome 16. So there's some similarity of humans. Localization of VKOR to 190 genes within human chromosome 16 narrowed the search to 13 genes encoding a candidate transmembrane proteins. So they used a very classic approach here. That's why it relates to the chapters 11, 12, 13, where first there was identification of the rat, and then using that information identify where it would be in humans, what's similar to it, and then looking at the genes there, which would be with bioinformatics analysis, saying when the genome being sequenced, determining which genes were reasonable, has to be transmembrane apparently, and we use short interfering RNAs pools against individual genes to test their ability to inhibit VKOR activity. So we had an assay for whether VKOR is there, and then one by one knocking out the candidate genes until it's seen. Here we report, okay, so that's then phenomenon of interest, specific background, and then what they did. First finding that they localized it, and we used this approach, and we found this. Here, now their findings very clearly, we report the identification of the gene for VKOR based on specific inhibition of VKOR activity by single siRNA pool. We confirmed that NGC11276 messenger RNA, which we can locate by the human genome, we can find it, encodes VKOR through its expression in insect cells. So they cloned it, put it in a different cell type, and sensitivity to warfarin, and found that then they could detect it experimentally. The expressed enzyme is 163 amino acids long, so relatively small, with at least one transmembrane domain, which is apparently expected. Okay, those are the findings. Now it's going to be significance. Identification of the VKOR gene extends our understanding of blood clotting and should facilitate development of new anticoagulant drugs. And so this is very brief, and I want to contrast that with this other paper by a different group, title here, Mutations in VKORC1 cause warfarin resistance and multiple coagulation factor deficiency type 2. So they're relating a specific gene to a human gene syndrome. So let's do this first paragraph. Let's read this. Coumarin derivatives such as warfarin, so it's a coumarin derivative, and there are other ones that are similar, represent the therapy of choice. So the other one stated is most widely prescribed. Similar idea, very different way of expressing it. For the long-term treatment and prevention of thromboembolic events. Coumarins target blood coagulation by inhibiting the vitamin K epoxide reductase multiprotein complex VKOR. The complex recycles vitamin K2,3 epoxide to vitamin K hydroquinone, so this is a reduction oxidation. A cofactor that is essential for the post-transcriptional gamma carboxylation of several blood coagulation factors. So far, they have told us the wide phenomenon, starting with the molecule, specific issues that we need to know in order to understand the significance and what they're talking about. Now, the question or the puzzle or the issue, despite extensive efforts the components of VKOR complex have not been identified. So that's the problem. The complex has been proposed to be involved in 2 heritable human diseases. So 2 heritable diseases have been identified or proposed to be related. One is combined deficiency of vitamin K dependent clotting factors type 2. So you can imagine that if this enzyme exists, we might expect that there is a null mutant of it or under expression or under activity, and indeed there is one, and there's an online Mendelian inheritance of man that's covered in the book. It's quite interesting. There's lots of ideas there, and you might pay attention to how many genes are identified and how many genes exist in the human genome, how many are Mendelian, and how many who have a mechanism of action that's understood, which is quite small. And resistance and the location, it's a OMIM number, and resistance to coumarin type anticoagulant drugs. So we have then, that would be a loss of function mutation that causes this deficiency of the VKOR, and here resistance to anticoagulant drugs. So that's a gain of function in which the some people have a mutation, which means that warfarin doesn't work to inhibit them, and this then would be what the rats, warfarin resistant rats, have also developed. And so that's something that has to be known. If we're going to treat somebody with warfarin for blood thinning, or it's very important to know whether or not it works or whether it works too much. Now the first group is some sort of probably hemophilia that's treated by possibly constant ingestion of fresh vitamin K or something. I don't know. We could read about it. And the second one would be, it turns out when their blood thinners are given to the patient that they don't work. Here we identify, so that's the significance, and here the telling us our findings. Here we identify by linkage information from 3 species the gene vitamin K epoxide reductase complex subunit 1, VKORC1, which encode a small transmembrane protein of the endoplasmic reticulum. VKORC1 contains missense mutations in both human disorders. So the missense means it's not a stop codon, it's a change in amino acid which causes something either to not work as well or to be resistant. And in warfarin resistant rat strain, that would be the gain of function, the one that they're resistant to. Overexpression of wild type VKORC1, but not VKORC1 carrying the VKORCDF2 mutation. That's the one that's underexpressed, leads to a marked increase in VKOR activity, which is sensitive to warfarin inhibition. So they're not describing very clearly the significance. They're very tight here. They're telling us here what their findings were and what can be done with it. And they're showing that they've identified the gene such that, and overexpression of wild type fixes it. So they're giving us their evidence, and they're saving a longer discussion of the significance for the body of this report. So we see these abstracts have a lot of information, but it's presented in a very brief, terse way because they're trying to pack in as much as they possibly can. And in this case, 2 different groups pretty much discovered the same thing by very, overlapping methods, but different methods and different strategies. And they express their findings in very different ways. So that's a good contrast. I will stop here.